Michigan. Dr. Gudjonsson did his MD and PhD from University of Iceland in Iceland. And then he joined University of Michigan as an intern. And then he did his residency in dermatology and joined the faculty in 2008 joined University of Michigan as a faculty in 2008. Dr. Gudjonsson leads an NIH-funded research group examining the immunological and genetic drivers of inflammatory skin disease. I think, aimed at improving the diagnosis and the treatment of various skin disorders. He has published over 200 peer-reviewed papers in top-tier journals, including Journal of Investigative Dermatology, Science Translational Medicine, Nature Genetics, Nature Immunology. He's very widely cited, highly recognized in the field. He has won many awards and accolades, and among them are the Young Investigator Award from the American Academy of Dermatology. He was named the 2018 Rising Star Lecturer for the Society for Investigative Dermatology and was recipient of the American Skin Association Research Achievement Award in Psoriasis in 2021. And his work has earned several research awards from NIH and also other foundations like American Skin Association, Doris Duke Foundation. Without much ado, now I would like to invite Dr. Gudjonsson to present his research on single cell and spatial analysis in inflammatory skin disease. Thank you very much.
Yeah, thank you, Sunita, for that very nice and detailed introduction. I'm very excited to be with you all today, and thanks again for inviting me to speak. I hope you can all see my screen, right?
Yes, very much.
Oops.
Nice.
Sorry?
Correct. Yeah, we could see.
Absolutely can.
I lost control there for like a second. I was trying to find the oops. So you can see the arrow also, right?
Yes, we could see your arrow, and also the screen is perfect in presentation format.
Again, thanks so much for inviting me to talk. I'm very excited to kind of be here with you today. And what I'm going to show you is actually how we used single cell and also spatial-seq technologies to kind of deconstruct an entire disease, systemic sclerosis. So that's going to be the focus of my talk. My lab has been doing more and more single cell sequencing and spatial-seq integration over the past few years, and been really sold on this tool as a way to really get very deep insight into disease mechanisms. So as mentioned before, I'm Johann Gudjonsson. I'm a dermatologist, and I've been here for 19 years. And my lab kind of focuses on chronic inflammatory skin diseases. So just to kind of give you a brief introduction of what scleroderma is. So it's a complex disease in which the skin kind of tightens up and hardens. And so it has extensive fibrosis. As you can see over here, the fingers kind of get contracted, the skin becomes shiny and pebbly, and it's actually it's rock hard. These patients also have vascular alterations, and they often have disturbances in the blood flow to the hands and the toes. And they can kind of get these ulcers like you can see over here, often from ischemia, and particularly here on the tip of the fingers. And it's a disease that is characterized by various autoantibodies. So there's a very strong indication that it's an autoimmune disease, but the mechanisms involved have not been fully cleared. It's a pretty uncommon disease, so the incidence ranges from 2.3 to 22.8 cases per 1 million persons per year. The genetics behind this are complex, so multiple different genetic variants, each one with a fairly small risk increase. And it's one of those diseases that have are more common in women. And the ratio ranges from 3:1 to 14 against 1. So a very heavy female skewing. And that actually is one of the reasons why my group got involved in studying this disease. We've been very interested in sex biases and autoimmunity. As I mentioned before, these are kind of classical pictures of patients with scleroderma. Some of these are photographs that I've taken from my clinic. And what you can see here, this is just the histology of systemic sclerosis skin. So what I want to highlight here is the hallmark of the disease. It's this very dense collagen matrix that is in the dermis. This is the key feature of systemic sclerosis: loss of adnexal structures such as hair follicles and sweat glands, and again, very dense extracellular component matrix. The pathogenesis of this disease is incompletely understood, but a lot of the research that has been done in the past 10, 15 years has focused on myofibroblast as a major source of the fibrosis in systemic sclerosis skin. So myofibroblast are a differentiated type of a cell. They arise from fibroblasts that can undergo this proto-myofibroblast in tissues. This is a cell that you can find under normal physiological circumstances, such as during wound healing, and these cells play a critical role in wound contraction and also kind of organizing the healing. But in wound healing, these cells kind of go away. But in systemic sclerosis, these cells kind of stick around, and they produce a lot of extracellular matrix components such as collagen. And the key feature that characterizes these cells is this dense matrix of alpha smooth muscle actin, and that's a marker that we often use to kind of identify the cell. So what we have done, we kind of got interested in this disease about 2 or 3 years ago. I was working with my colleague in rheumatology, Dr. Dinesh Khanna, who is a scleroderma expert, and we decided to kind of look at this disease more closely with single cell and spatial-seq technologies. So these are data, single cell data, that I'm sure you're all familiar with. This is from 26 systemic sclerosis patients with early diffuse disease, and then about 18 healthy controls. So you can appreciate this is a UMAP. You can appreciate the different cell populations that we capture in our single cell data. So we have a very good representation of both the different stromal cells, such as keratinocytes, fibroblasts, hairy-cytes, endothelial cells, and then various immune cell subsets. And this kind of shows you the shifts that you can see in the disease process from normal skin, shown in blue, towards systemic sclerosis. So you can see that these do not perfectly align, and that is pretty evident in all the different cell populations that you capture in scleroderma skin. And particularly here in the fibroblast, this kind of a tail over here is entirely in systemic sclerosis fibroblast. Major shifts in some cell populations, so mostly in smooth muscle and then B cells, and that's characteristic of systemic sclerosis skin, but otherwise a fairly even distribution across all the cell populations. And this is just the top 3 marker genes for each cell cluster that we identified in our UMAP, and kind of gives you an idea that the distinguishing, well, fairly easy to distinguish between the different cell types using these markers. So one thing that we have done is to kind of use spatial sequencing to really gain a better idea of what's going on in the tissue. That's always been the limitation of single cell sequencing. It really doesn't give you an idea where things are happening in the sample that you have. So one thing that we have been doing is to kind of integrate the two, the single cell with the spatial-seq. So what we do is we use the single cell data as a signature for the cell population in skin, and then we cross-analyze that against our spatial coordinates. And this is the Vision 10x system. And obviously with a 55 micrometer resolution of each dot, it's kind of hard to kind of nail it down to a single cell. But by using these tools, we can actually have a pretty good idea where some of these signals are coming from. So you can see the keratinocytes are lining along the top of the biopsy where you have the skin cells, and also around the hair follicle. The melanocytes are scattered here on the dermal epidermal junction. The acron glands are located deep in the skin, so we're capturing these pretty well. And you can see fibroblasts more diffuse through the skin, the pericytes, the cells around the blood vessels, the same thing. Smooth muscles, you get them more dense around the hair follicle, and then you get inflammatory cell infiltration, such as T cells, myeloid, so mast cells. You get diffuse staining of or presentation of endothelial cells, nerve cells, and then clusters of B cells in the skin. So by just doing the spatial single cell integration, you can actually capture a lot of the cells that are known to be involved in systemic sclerosis skin. Obviously, you need to validate this by immunohistochemistry, but again, the alignment when we have done this has actually been pretty good. One way to kind of use these data, particularly in terms of the cell types, is to look at receptor ligand interactions to kind of get an idea what cells might be the ones driving the disease process, and particularly in terms of cell-cell communication. So we're looking at the receptor ligand pairs that are increased in systemic sclerosis skin. And we have up to 120 pairs here shown in red. When you look at it this way, you can see that almost the great majority of the interactions that are happening in systemic sclerosis skin are happening between only 3 specific cell populations: endothelial cells, fibroblasts, and myeloid cells, both in terms of the ligands and the receptors. Obviously, more things going on, but that's the key population that is driving most of the communication. And we can also look at it this way by looking at the interactions in this kind of a structured kind of an analysis, and you can really see that it's the fibroblast, myeloid cells, and endothelial cells that are sticking out, but obviously a lot of all the communications going on with all the cell subsets. But it's a very useful way to really prioritize which cell types to kind of focus on. And again, with the fibrosis being the key histologic feature of scleroderma, we kind of are focused mostly on the fibroblast. These are the subclusters of the fibroblast that we identify in healthy and scleroderma skin. So we have 7 different subpopulations of fibroblasts that we have kind of labeled with the top gene in each class, and many of these correspond with other fibroblast populations that have been previously studied and described in skin. And this kind of gives you an appreciation of the shifts that are happening in systemic sclerosis skin. So you have the normal fibroblast here in blue, and then you have the systemic sclerosis fibroblast shown here in red. You can see that there's a lot of shifts happening and some populations that are unique to systemic sclerosis skin, such as the collagen 8A1 positive fibroblast. And this is a population that actually represents myofibroblasts. And is only found in systemic sclerosis skin. But there are other populations such as pro-inflammatory fibroblasts that are also increased in systemic sclerosis skin. So one thing that we have done is to kind of look at which one of those fibroblasts are the ones that are most active in terms of deposition of extracellular matrix component, given that that's the key feature of the disease process. And when we do that, we use this extracellular matrix module score. And then we look at that in the different fibroblast populations. So what you can see here is that there are 2 populations that stand out: the SFRP2 positive fibroblasts that are only here in red, and then the collagen 8A1 positive fibroblast, which represents the myofibroblast. So this cluster right here is the one that is responsible for most of the extracellular matrix component production. We can look at the marker genes over here, and I want to highlight that some of the collagens are genes are most prominent in this population over here. And then we can use the spatial-seq data again to really see where the fibrosis is happening and how does that correlate with those different fibroblast populations. So you have collagen 1A1, which is a key component of type 1 collagen. By looking at the expression of that on the spatial-seq, this is 4 different patients with scleroderma that we have over here. You can really appreciate that there are areas in the tissue where there's much more going on than other places in the biopsy. So the fibrosis is not happening throughout the tissue, it's actually focal. You can see that here, same over here, and even this one over here, there are hot spots, and then there are spots where there's minimal to no activity going on. You can also overlay the module score, and that will give you even kind of a finer and more granular view of what's going on, highlighting better these areas where there's more of that profibrotic activity going on, and correlating to some extent to collagen 1A1, as you would expect, and the same over here. And then you can take these 2 populations, the SFRP2 positive fibroblast and the collagen 8A1 or myofibroblast, and see where they're located in the skin. So the SFRP2 are mostly here in the epidermis, whereas the myofibroblasts are much more deeper in the dermis along the dermal subcutaneous junction. And this fits with what's been described in scleroderma before, that the myofibroblasts are mostly found deeper in the skin. So looking at smooth muscle actin and using immunohistochemistry as a way to kind of validate it, you can see a lot of the smooth muscle actin in the epidermis is around the blood vessels, whereas the majority of it in the deeper layers of the skin are diffusely on fibroblasts. And we can do the same thing here, looking at it by immunofluorescence, looking at the co-localization of mymentin and ACTA2. The ACTA2 is around the blood vessels. Most of the fibroblasts in the superficial dermis do not have smooth muscle actin, so they're not myofibroblasts, but the great majority of the fibroblasts deep in the skin or deep in the dermis are myofibroblasts. in origin. So it kind of gives you the spatial context of what you're actually looking at. You can also do the same thing with all the fibroblast subsets. So here we're looking at the inflammatory fibroblast, the CCL19, CLDN or Claudin 1 fibroblast, FMO1, FMO2, and TNN fibroblast. And you can see how much they're much more diffuse and evenly distributed over the skin. These are the different clusters that we have. Again, these are the 7 fibroblast populations that we have looked at, but the ones that kind of caught our eyes and we kind of got focused on are the SFRP2 positive and the collagen 81 positive of the myofibroblast. Particularly given this is looking at all the different collagens that are produced in skin, and these are the different fibroblast populations in scleroderma skin. But it's only these 2 that are really producing the type 1 and type 3 collagen that is so enriched in systemic sclerosis and drives the skin hardening that you can see in this disease. So that's the subset that we kind of focused on more deeply. And that's kind of shown over here when we're looking at the SFRP2 and the myofibroblast. You can see these are actually, we can distinguish those into 3 different groups. These shows you again the shifts from normal skin to systemic sclerosis. And then we can look at specific marker genes, such as smooth muscle actin 2, mostly found here in the collagen 81 group, which are the myofibroblast. Transgelin, again, showing you the same, and then the collagen 81, which is our marker that we use to define the subset. So you can really appreciate that there's a transition from this over here. If you look at the extracellular component module, it kind of goes up as you move along the trajectory from SFRP2 positive to the myofibroblast. Same with TGF-beta, Th2 signal, and then also hippocomponent genes such as connective tissue growth factor and CYR61. And one thing that we have been using quite a bit is this signature called cytotrace. We find that to be quite useful. It's a way to kind of measure the differentiation status of the cells. And you can see here that as you move from this cluster over here, that is in normal skin, and as we move towards where the myofibroblasts are, the color is getting more intense. So that to us suggests that there's a differentiation trajectory that is going on, and you're kind of moving from less differentiated to more differentiated cells. Why is that helpful and why is that important? What it does, it actually gives you an idea of what is the direction of your pseudotime. If you use a pseudotime to kind of go across and kind of tell you what is the transition, you would expect that trajectory to go from less differentiated to more differentiated fibroblast. And that's and therefore the direction of the change. So this is the pseudotime that we drafted. It's a single kind of a curve, and we can align the different groups along the pseudotime trajectory going from group 1 to group 2 to group 3. And the direction is likely this way given the cytotrace signature. But what we can do is we can then use upstream regulator analysis. This is something, it's a function of the Ingenuity pathway analysis tool. Don't find IPA that useful, but for this, it actually can be very helpful. So we use that tool to really tell us, with the gene expression changes that are happening along the trajectory, what might be upstream of it and driving it. And then we look at the correlation across the pseudotime or module score. So you can see the TGF-beta 1 is getting increased, the SMAD3 is also getting increased. We see STAT1, which is indicative of an interference signature, also being increased. To a lesser extent, IRF7. RUNX1, which is a risk factor for systemic sclerosis, actually is increased as you move towards the myofibroblast. Th2 response, and then component of the HIPA pathway. This is WWTR1, which is another name for TAS, and then the T1 transcription factor. So again, it can give you an idea of what might be driving the shift towards myofibroblast. So again, this is the pseudotime trajectory. We can kind of trace the gene expression across the different the patterns as they kind of move from regular fibroblast to myofibroblast. And then again, we have been, given that the TEAT pathway or the HIPA pathway seem to be playing such a critical role, we've been looking at different components of the pathway as you move from regular fibroblast to myofibroblast. It's particularly the different TEATs, the TEAT1, TEAT2, TEAT3, and TEAT4 that seem to be highly enriched in the myofibroblast population compared to the other two. And that kind of made us intrigued and interested in figuring out, what is the role of the Hippo pathway in kind of driving this process. So this is a complex schematic, but I really wanted to show you what the Hippo pathway kind of looks like. It's a complex pathway with multiple different regulators. The key pathways is this YAP-TAZ. YAP-TAZ can be phosphorylated and then retained in the nucleus, and that is degraded. But it can also be shifted into the nucleus where it binds the TEADs. And when it binds the TEADs, it actually drives gene expression program that actually is pro-inflammatory and pro-fibrotic. When we use an inhibitor here called Truly, and I'm going to show you some data on that, we actually inhibit LATs. You don't phosphorylate YAP anymore, and you shift the YAP into the nucleus where it can kind of drive fibrosis. If you use another inhibitor called verteporfin that inhibits the interaction between the YAP and the TEAD, you can shut down the activity in the nucleus and therefore shut down the profibrotic response. So if you use myofibroblasts and we expose them to Truly or verteporfin, we get very dramatic changes in terms of the ACTA2 expression, the smooth muscle actin, or collagen 1A1 expression. So Truly, by shifting the YAP into the nucleus, will increase smooth muscle actin and also collagen 1 expression. If we inhibit the interaction at the transcription factor level, we can really shut down ACTA2 or collagen 1 expression. And we can validate that by protein, like shown over here, and then also by these functional assays. So this is a gel contraction or gel relaxation assay. So if you use Truly, we actually tighten the gel kind of shrinks because you're getting more smooth muscle actin and it's shrinking it down. Sorry. Any questions? Well, and then you can also use gel relaxation assays that kind of tells you how the gel is kind of relaxing. And you can see that we are getting the opposite with verteporfin. So with Truly, by activating this pathway, we get contraction. By using verteporfin that inhibits this, we get relaxation of the assay. And again, that kind of aligns with the fibrotic process that is going on in the skin. We can also look at the confluence. We're getting increased growth of fibroblasts with Truly, whereas we're getting the opposite with verteporfin, where it actually inhibits myofibroblast growth. We can also look at it in terms of scratch assays, where we're looking at wound confluence, with the Truly and the verteporfin have opposite effects. So by modulating the Hippo pathway, we can really shift the whole process away from myofibroblast and away from fibrosis. And again, this is siRNA validation of the inhibitor assays, showing that by silencing YAP1 or TEAD1, and to some extent T3, we can shut down ACTA2 expression, the same with collagen 1A1 expression. And this kind of shows you an immunofluorescence of that. This is looking at smooth muscle actin. This is collagen 1 expression, so you can see that there's very high expression of that in baseline myofibroblasts from systemic sclerosis. And by adding Truly, we actually get more, but by adding or inhibiting the YAP-TAZ or YAP-TAZ TEAD interactions, we can really shut down collagen 1 expression. And same thing with the smooth muscle actin, which is characteristic of myofibroblast. By having Truly, we get a lot more of it. By using verteporfin, we can really almost shut it down. So what about all the cell types? So as I showed you before, endothelial cells seem to be playing a critical role in systemic sclerosis as well. We capture 7 different endothelial clusters in systemic sclerosis. You have the lymphatic ones over here. And then you have this kind of extended cluster over here, 5, 4, and 3, and 0, 1, and 2. And if you look at the shifts that are happening in systemic sclerosis, again, you can see these marked shifts shown here in red, both in this endothelial population over here, and then also down here. And by looking at the percentage or proportion in terms of the shifts, as we're going to go through the disease states, it really is the population number 2 that is here, and population number 4 that is over here, that are really the most dramatically changed populations in endothelial cells and systemic sclerosis skin. We got really interested in this population number 2 here, because again, it actually is expressing a lot of the same collagen genes that we saw in the myofibroblasts. Another thing that kind of characterizes this set over here is the gap junction A4. This is actually a marker. It's a connexin, so it kind of provides a connection between different cell types. It's only expressed in the first three subsets, so 0, 1, and 2, which is this one over here. This is a marker for pre-arteriolar endothelial cells. And the reason why we kind of got interested about this one is that many of the ischemic changes and the vascular changes that are happening in systemic sclerosis skin are thought to be in the small arteriolar blood vessels that are kind of feeding into the skin. So this aligns well with where the pathology of the blood flow pathology is in systemic sclerosis. And this subset was very interesting, given that it was pro-fibrotic. So if you look at endothelial markers, so this is PCAM1, which is CD31, we can see that it's present in all three subclusters over here. Vascular cadherin and cadherin 5, same thing, but it's decreasing as we kind of move down this path. The Notch signaling pathway, Notch1, JAK2, and DLL4 kind of goes down. We have expression of collagen genes, but it's mostly the collagen 1A1, transgelin, and ACTA, or smooth muscle actin, that is much more highly expressed in this cluster over here and again, that's fairly highly enriched in just systemic sclerosis skin. And again, we see the same thing over here. This is just a feature plot showing the expression of CD31, V-cadherin, lymphatic markers, P-selectin and E-selectin, and then this gap junction A4, which is the pre-arteriolar type blood marker, and then smooth muscle actin and collagen A1 to kind of give you an idea of the expression of it. And again, it's the subset 2 that is very pro-fibrotic with very strong expression of type 1 and type 3 collagens. And if you compare these endothelial cell populations to the main fibroblast populations that are pro-fibrotic and systemic sclerosis, we see that this endothelial subset produces quite a bit of type 1 collagen, although not quite as high as the SFRB2 or the myofibroblastic systemic sclerosis skin. We can look at it, more deeply. This is the extracellular component module. We can see that that's very high in the cluster 2. We use the cytotrace to kind of look at the differentiation status of these cells, and you can really appreciate how high it is in this cluster 2. So it kind of sets that if you're doing a pseudotime trajectory against these three clusters, it will kind of tell you the direction of the change. This is the pseudotime, again, a kind of single trajectory. And then the clusters kind of go from 0, 1, and 2, and that aligns almost perfectly along the pseudotime trajectory. Similar as we saw with the myofibroblast. So this is the fibroblast to myofibroblast transition, and this is looking at the correlation coefficient against the pseudotime. We're seeing that there's an alignment between the endothelial and the endo-MT transition and the fibroblast myofibroblast transition that centers around the HIPPO pathway and the TGF-beta pathway. The interferon pathway that I highlighted and showed you before with the myofibroblast population is only found in the myofibroblast. We do not see that in the endothelial cell population. And if we look at the same HIPPO pathway components, you can really appreciate that many of them are much more highly expressed in this cluster down here than in the other two clusters. That includes the YAP, TAS, CTGF, CYR61, also the sex-biased transcriptional regulator that we identified a few years ago, VGL3, very selective for this population over here. And then the 4 TEAT members, the transcription factors, very selective and very highly expressed in the endo-MT population. We validated this by immunofluorescence, so we can really appreciate the endo-MTs aligning with the blood vessels in scleroderma skin. We do not really capture these that much in healthy skin. And then we have also validated this by immunohistochemistry against the different components in both systemic sclerosis skin and also against healthy controls. And this shows you the inhibitor assay that we have done. It's pretty much the same thing that we did with the myofibroblast. So if you look at ACTA2 or smooth muscle actin expression, by adding in Truly, that shifts the YAP into the nucleus, we get much more of it. Whereas if you use verteporfin that blocks the TEATs, we can really shut it down. Same thing for collagen 1A1 expression, Truly increases it, versus the verteporfin kind of shuts it down. And we can also see that these same markers also affect vascular differentiation to some extent, by affecting CD31 and VCAM expression. We validated this by immunofluorescence, Western blot, and also by immunofluorescence. So this is looking at endo-MTs and systemic sclerosis skin. If we add truly, these cells kind of become much more spindly, and we get a lot more smooth muscle actin. And if we use verteporfin, we can really get rid of the endo-MT phenotype. But we get more strongly kind of induction of the vascular phenotype shown here with the 1 milligram factor staining. And this is a close-up where you can see the smooth muscle actin and the endothelial markers. And if you really activate this pathway by using truly, you get much more endo-MT transition. But if you use the verteporfin, you can practically shut it down. So lastly, I want to kind of show one thing that is actually quite useful is that when you have kind of gone through that, it's not just to look at the main cell subtypes that are kind of interactive, but also the cell subsets. And when you do that, obviously you get a very complex kind of interaction kind of map of what's going on in systemic sclerosis. But it's very interesting that the major cell interactions that we see in systemic sclerosis is actually happening between the myofibroblasts shown over here and these endothelial cell 2 or the endo-MT subpopulation. There's also quite striking interaction with profibrotic fibroblasts, but also the other endothelial cell subsets actually seem to be quite dynamic and contributing. And also some of the myeloid subsets, such as the conventional dendritic cell 1, conventional dendritic cell 2b, and 2a. So it kind of gives you an idea, and a path where to kind of dig down into the deeper into the biology. And this is also a very useful way that we have found to kind of really highlight those different receptor ligand interactions. So here we're kind of looking at the different endothelial subsets, and here we have the fibroblast subsets. Here we have a selection of the ligands that are active and found in myofibroblast and systemic sclerosis. And then you can match that up with the different receptors that are kind of found on these same subsets. So again, it can be very useful. It kind of gives you an idea how much more you have of this in the myofibroblast. And some of these key pathways are what have been targeted with therapeutics recently, such as IL-6. You can see IL-6 is almost entirely from the myofibroblast population, and then acting here on other fibroblast subsets. So it gives you a way to really dissect and see how these cells are kind of communicating, and what is also the key population in this disease process. So to kind of briefly summarize, so these data demonstrate the power of single cell and spatial-seq technologies, and particularly the integration of it to unravel pathogenic mechanisms in scleroderma. So cellular interactions are primarily between only 3 cell types: the myeloid, endothelial, and fibroblast. And these data, particularly when combined with the spatial-seq aspects, it really demonstrates compartmentalization of profibrotic processes and also differences in spatial content of specific fibroblast subpopulations. And then what we show here is that scleroderma really has a dual source of collagen and extracellular matrix producing cells in skin. And that is both myofibroblast and pro-myofibroblast or pre-myofibroblast, and also this endo-MT cell population. And again, we show how that is distinct in different layers of the skin, and then also how we can use these data to really get the upstream regulators that really are driving the disease process, such as we did with Hippo pathway, and that it kind of drives not just the myofibroblast, but also the endo-MT transition in systemic sclerosis. So that concludes my talk. I just want to kind of acknowledge some of my key colleagues that have been instrumental in putting this together, particularly Dr. Kanna, Dinesh Kanna, John Varka, Fei-Yang Ma, who has been is our bioinformatic person that has been overseeing our spatial and single cell analyses, and also Alex Soy, who has been instrumental in that. Alison Billy, a colleague that has been very useful and helpful in kind of pulling this together, and my colleague also, Dr. Kallenberg, and Eliza Su, who has helped us with the in vitro experiments, and then also outside collaborators. And last but not least, the various sources of funding that I've had over the years that have helped support all this work. So thank you for listening, and I'm happy to take questions.
Thank you, Johan, for that wonderful and very detailed talk, very exciting work. Now, this is open for any question and answers. Please feel free to unmute yourself and ask a question, or type it in the chat window, however you feel comfortable. Now the session is open for any Q&A, please.
Johann, this is Pierre, your colleague from dermatology and biology. Wonderful talk. I wonder whether you would comment on the involvement of other tissues that have a similar connective tissue matrix as the skin, or is scleroderma, and pardon my lack of knowledge, is scleroderma a primarily skin disorder?
No, that's a good question. It actually is a systemic disease, and what usually it can be deadly and a very high. It's actually one of the deadliest autoimmune disease that you can actually have, and the reason for the death is actually lung fibrosis and then also kidney involvement. So it truly is a systemic inflammatory disease, but the link between the different tissues is not really known. But there's certainly similarities in terms of the disease process that are going on. So I think the pulmonary fibrosis and probably the renal issues are probably all related to the same thing that we're seeing in the skin. And in terms of the tissue, if you can make single cell sequencing work on skin, you can practically make it work on any tissue. The skin is like chewing gum. It's incredibly tough to get through. And we've used the protocol that we've used to kind of digest the skin and get these single cell populations. We've used that on all the tissues, such as blood vessels, aortic tissue, so like heavily fibrotic tissue, and it actually works really, really well. So again, that really shouldn't. I think probably bone might be difficult, but that's the only tissue. I haven't really tried that yet.
Thank you.
I have a question just to follow up what Pierre asked. I was just wondering, what are the learnings from this to other autoimmune skin-related diseases like vitiligo? Like,
Yeah, that's actually is really interesting. We've been looking at a range of other inflammatory skin diseases such as psoriasis and lupus and dermatomyositis and hidradenitis. And it really highlights the differences, the single cell. We see sometimes similar cells. I see like myofibroblast in like diseases such as hidradenitis. We actually have fibrosis, but they're a little bit different. So I really think that the single cell seq can really help you appreciate the plasticity of the immune response that can happen in a given tissue, and it's shaped by so many different factors that the end outcome is different, and we see that in the single cell data. So I've never come across any technology that really gives you the ability to really dig this deep in, but I think it's hard to extrapolate from one disease to the next. So that's the simple answer. I think you really need to go in there and get a sample, because again, even if it is a similar process with your fibrosis, there's still very prominent differences.
Yeah, sounds good. Thank you. And did you see any limitations while others are asking? Just, it's very exciting talk. I answered another technical question about the resolution you got around with the vision, maybe like 50, 60 microns. Do you think, do you missed anything? Is it limited because of that? If you had a 10 micron resolution spatial analysis, will your data would look different? Or is it going to be better? Or in this case, it didn't matter?
No, it definitely matters. You wonder what you're missing, and it's not just, it's like 55 micrometer is the Vision 10X system. And in each dot, you can assume that you probably are capturing like 10 to 20 different cell types, different cells. And when the cell types are all the same, it actually becomes easy to kind of deconvolute it with the signatures. But if it is a mixed population, it can be quite hard. And we've used like pie charts to kind of address that. We have like, you can attribute like 60% of the signature to myeloid cells and maybe 20 or 30% to T cells, and then 10% to endothelial cells. But it's by no means perfect. And then also, it's not that it's just 55 micrometer dot. There's also a gap between them. And you wonder what you're missing there. And you know, there are cells there that you would like to capture. We have worked with Johann E. He actually has the SCSAP scope, and I think that's a very powerful way to kind of look at that. That kind of potentially gives you like a subcellular localization. The problem is that when it drills down that deep, is that the information that you get per dot is going to be much less than you get at the 55 micrometer resolution. So it's like there's always this kind of a trade-off. But I do think that when the Vision comes out with a high definition system, that probably will happen this summer. I think that's going to change things a lot, and that's when the spatial-seq, you can practically start manipulating that data like single-cell-seq data. And who knows, it might even make single-cell-seq obsolete. You know, I think spatial context is critical.
Yes, thank you.
Hello, I have a question. A beautiful talks. I am from endocrinology. I'm interested in adipose tissue biology, and wondering, in mice, so you have so-called hypodermal adipose tissue, kind of close to the skin. It might be different from humans. So, but question, I have two questions. One is any role for pre-adipocyte or adipocyte for that your ECM-related gene expression. Second question is, if you have the skin mouse model of your scleroderma, do you see the significant difference between humans and mice? And if there is any difference or not, but how we can extrapolate or use that mouse model to understand human disease model?
Yeah, great question. You know, the one of the limitations of the regular single-cell-seq is that it doesn't allow you to capture fat cells. We see that a lot, and it's like one of the populations that I really miss not capturing. There's also another population of skin that you don't capture, and that's the oil glands, the sebaceous glands. The same issue. They're too big and they float. It's kind of not optimal to miss them. There is some evidence in systemic sclerosis of some of the myofibroblast population might be arising from adipocytes, and maybe that's one of the reasons why they seem to arise deep in the dermis and not so much on the surface. The only way to kind of capture that is to do a single nuclear sequencing, and we're doing it. And again, it's just a little bit of a different processing, but it works really well. In terms of the animal models, the limitation there are just the data are not available. And I was looking at it, like the bleomycin mouse model is like the classic one that they use in scleroderma, where they take bleomycin, just a chemo agent, they inject it into the skin of the mice repeatedly, and then the mice actually develop fibrosis. But even with that disease process, it's not even clear if it is like a myofibroblast-driven disease process that these mice get. And nobody has really done the cross-species analysis to see where those biological processes actually align. Because I've done a lot of work on psoriasis mouse models, and I find, I think these mouse models are useful. They definitely model the disease process, but you have to know where they align. And if you don't have that information, it's very difficult to kind of take them as the entire disease, because they don't. They only model like very certain and limited aspects of the disease process. So I tend to be very selective. We are doing the bleomycin. We're actually going to do single cell sequencing of it. So I think we will be able to answer it. So I think, the mouse models are partially useful, at least the mouse models.
Tom, do you have a question? I see.
Yeah, I just was going to ask a couple of questions that came back to sort of the beginning. So first of all, it's a really beautiful presentation about integrating the single cell and the spatial information. And yeah, we definitely want to see some of the resolution issues pushing forward. Yeah. You started out by discussing the sex bias in the disease. And so that's two questions back to back. One is how much this single cell and spatial data did they help inform that question? And then the second one is just a very general naive question perhaps for me, which is that in terms of treating the disease, do you have a sensibility that if you had a pharmacological intervention that came out of these pathway discoveries, could it be used to reverse the disease, or would it have to be preventive?
Yeah, great question. No, the sex bias actually came from a previous work that we did before, and that actually was mostly out of bulk RNA-seq. We just asked a simple question: what's the difference between male and female skin? We identified a few genes, and one of them was this transcription cofactor called VGL3. And if we overexpress that in mouse skin, we actually develop like a mouse, actually like an inflammatory phenotype that looks strikingly like lupus. And with autoantibodies and B cell expansion and B cell activation, and that's kind of the features that go across many of these sex bias diseases. We got intrigued by this because, again, this factor VGL3 regulates the Hippo pathway. It actually binds the TEADs. And when we looked at, in our data, the myofibroblast, and particularly the endo-MTs, the VGL3 factor is really, really high in both of those populations. So we don't have the data yet. It does seem to have some role, because if we knock it down, we do get less myofibroblast or less endo-MTs, and also less fibrosis. So it's kind of intriguing to kind of postulate that maybe the sex bias that is so striking in systemic sclerosis might be acting at that level, just regulating the Hippo pathway and regulating it differently in males versus females, maybe facilitating the switch in women. But we just don't know yet. But that's how we kind of got into the sex bias. I think single cell is obviously would be very powerful to address that, I think, in many ways. Particularly if you're looking at like X chromosome, effects, and you could combine that with like X special quantitative trait loci if you wanted to. So I think there's a lot of opportunity and potential. Just have to have high enough resolution data and probably genotype data at the same time. And your second question was again
Preventive versus reversible.
Yeah, it's a great question. Also, it's the problem with collagen is that in particular collagen in the skin, it has a half-life of like 15 years. So I think once it's out there, it's really hard to get it back. So I think preventive treatment is going to work the best. But that being said, it's not usually that it's just the collagen that is induced or increased in these diseases. It's also a lot of other extracellular matrix components, and that includes the MMPs. So you might think about that. There might be a way towards shifting it towards less production and more degradation also at the same time. So I think it's not impossible to reverse it to some extent, but I think the most likely approach is still going to be, the most successful approach is going to be preventive and treated early.
Thank you.
Hi, Johann, I have a question. This is Laura Cooney from Rheumatology.
Hi, Laura.
I was just wondering if you had a chance to do any longitudinal skin samples, because I think about T cell phenotypes sort of transitioning over time over the course of scleroderma, and I'm wondering if you see any consistent changes from early diffuse to later stages of disease.
No, great question. We are doing it. We're going to be doing it as a part of a clinical trial, actually, so where we have placebo controls. So we'll be able to address it. I would expect that we will see changes as the disease process kind of evolves. Without a doubt.
Great, thanks.
Hi, Johann. This is Venkat Keshamani from Pulmonary. I was wondering, so you talked about endo-EMT in your talk, but I was wondering the other potential source could be epithelial mediator or EMT mediated fibroblast sources. So I was wondering, did you see any epithelial components in your analysis or not at all? Epithelial
mesenchymal?
Yes.
No, we did not. We looked and we couldn't, at least we couldn't see a clear evidence for that. And we do it pretty crudely sometimes in terms of how we look for it, and we look for, even on the UMAP, if there's a connection between the subset where we can see that cells are kind of like in a transitional state. And when we don't see those, we don't really bother to kind of dig in deeper. We want to see some kind of a continuum. So we didn't really see it. We didn't really look that closely, but we didn't really appreciate it. whereas the endo-MT and the myofibroblast translation was very, very obvious.
Okay.
Any other questions?
Hi, Johann. My name's Alex Knight. I'm from the Department of Orthopedic Surgery. I really enjoyed your talk. Thank you. Fantastic stuff. I've got a technical question. How do you generate the ECM module score cluster by cluster?
Oh, it's just a signature. So it's like just a gene list.
Okay, so it's like a composite of all the collagens or something?
Yeah, it's more complex than that. I think we took it out of a publication. So that's where we got it. And then we have been using it. And
Okay.
again, if you want to get that module, I'm happy to get it to you.
Okay, great. I'll reach out to you. Thanks for that.
Yeah, and I think we will have a primary publication that will be able to cite, so it will make it, you know, more legit and easy to kind of use. We use these, it's almost like a gene set enrichment type analysis, but it's actually incredibly useful sometimes.
Okay, super. Thanks.
Any other questions? We have a couple of minutes towards the end of the hour here. Okay.